So let's get into touch and proprioception. These are the different topics we're going to be covering. We're going to be talking about different types of skin. So glabrous versus hairy skin. We're going to discuss briefly dermatomes and the implications of dermatomes, as well as the four major sensory cell types that are responsible for touch and proprioception. Then we're going to discuss receptive fields and the 2-point discrimination test. So we briefly discussed 2-point discrimination earlier on this semester when we were talking about like the cortical landscape in your brain and how much real estate is dedicated to different parts of your body. But there's also going to be cells that are implicated in that as well. So like certain cells you're going to find like in higher abundance in, for example, the tips of your fingers, right? So you'll get much more accuracy in the 2-point discrimination test. So there's different ways you can do that 2-point discrimination test. This device is one way. I've never personally used one of these, but that's one way you could do it, right? So super cool. The more detailed and granular that the information you receive, the better you are at predicting whether or not there's 2 different points touching the area, right? You will see that in higher concentrations in places like your hands, fingertips, and much less accuracy and concentration with like cell types and whatnot in areas like your torso, your thorax. Right? You're not going to be able to distinguish between those 2 different points. We're going to talk about different types of afferents, slow versus rapidly adapting, proprioception. We're going to discuss that in a little bit more detail. We've already talked about proprioception quite a bit. We'll go into some of the cell types in your muscles that are also involved in proprioception, as well as the muscle spindles, the different cell types that you find in muscles. We're going to discuss the Golgi tendon organ, which basically serves as a protective mechanism to prevent overstretching and strain on the muscles. We're going to talk about those ascending pathways in more detail now, in more detail than we did before. So the dorsal column, right, as well as the spinal thalamic, spinal cervical. We're also going to talk about the role of the thalamus, because that's going to be one of the major relaying centers for a lot of your afferent input. And then we're going to talk about the cortico communication, where you have different regions of your cortex communicating with each other. And then we're going to talk about a couple different diseases. So Friedreich's ataxia. We've already discussed Parkinson's quite a bit. We're going to go into Parkinson's in much more detail when we get into like the basal ganglia later on. And then we're going to cover a little bit of autistic spectrum disorder. Cool. So what does the skin do in terms of sensory input? So the skin is going to be responsible for a lot of your afferent input. And there's different cell types that are all specialized to be able to detect different types of touch. So some cells are going to be responsible for vibration. Some are responsible for more like rough touch, shearing type stress, stretching. Some cells are going to be associated with more of like a deeper type sensation. So different cells are going to be doing different types of things. We also have different skin types. When we talk about glabrous skin, that's what's on your hands, right? So the soles of your hands, the soles of your feet. You also find this on your lips too. These skin types are going to be extremely sensitive, right? Think about the lips, very sensitive. The tips of your fingers, very sensitive. So 2-point discrimination tests on these types of tissues are going to be a lot more fine-tuned, right? Much more granular in terms of detail. So, and this goes across species. It's not just humans, it's other animals too. This is a slide from a study that was looking at glabrous skin from different types of rodents. All sorts of different types of rodents. Rats, mice. I was comparing these images to the feet on my guinea pig. Very similar. My guinea pig's foot actually looks like this one over here on G. Looks like this mole rat feet. I know it's kind of gross. The big difference is that guinea pigs have 3 fingers versus these guys all have 5 fingers. So kind of cool. Hairy skin that's found basically everywhere else on your body, right? And you can see both discriminative and non-discriminative touch involved in hairy skin. It's not going to be as sensitive as glabrous skin, but it's still pretty sensitive, depending on which part of the body it is. You have a couple different types of, mechanoreceptors, which we're going to go into in a little bit, but one example would be low threshold mechanoreceptors. That's going to be due to innocuous stimuli. When we say innocuous, it means it's not painful, right? You can actually just feel whatever it is that you're feeling, but it's not causing any sort of pain. And then you also have dermatomes. And dermatomes are basically like a topographical map throughout your entire body. And these are going to be corresponding to this, the ganglia of afferents along your spinal cord. And because of that, if you have damage to a certain region on your spinal cord, you'd be able to see the corresponding region that's affected through the dermatomes. So if you did like a comprehensive neurological exam on a patient that had like a spinal cord injury, you'd be able to see the regions that maybe have some deficits in touch and other types of sensation as well. Pinprick sensation, touch sensation. So we'll go into that in a little bit more detail. Another thing too, with regards to cell types, is that you have, for pain and temperature cell types, this is going to be really big. We're going to talk about this in more detail as well when we get into like the pain and temperature section. But the cells that are going to be transmitting pain reception, those are going to have free nerve endings. Have you guys ever cut yourself and you, like, it hurts and then you put your hand in your mouth and it kind of like helps to alleviate the pain a little bit? It's almost like an innate reaction. You just kind of like do that right away when you hurt yourself. One of the reasons why that alleviates the pain is that those free nerve endings, those are exposed to air when you get a cut, and then that's one of the mechanisms how you start feeling pain. As soon as you like put your hand in your mouth or whatever, and you cover it and maybe a little bit of a saliva, that helps to alleviate some of that pain because you're basically covering up those free nerve endings. So we'll talk about that in a little bit. Other things that are important is that you have ganglia that are going to be receiving your afferents from the periphery. So these are going to be a series of dorsal root ganglia. Remember, your dorsal roots along your spinal cord are they're going to be like the receiving end of your afferents. And then when you go to the head, so head sensory input that's going to be coming in through the trigeminal nerve is going to be coming specifically through the trigeminal ganglia. So everything besides the head and face and all of that, when it comes to sensory input, it's going to be from the cervical, thoracic, lumbar, sacral ganglia throughout the entire spinal cord, with the exception of the head. So any sort of sensory input will be coming through the trigeminal ganglia. So make sure you know that. Can you say it again? So if you were to like touch your face, right? So remember your trigeminal nerve, cranial nerve what? 5, right? Cranial nerve 5, whenever you're getting sensory input, that's the trigeminal nerve. You're going to get sensory input going in through the trigeminal ganglia. So it's going to be making its way basically to the brainstem. Whereas everything else, in terms of sensory input, which I'm going to show you the dermatomes in a second, those are going to be coming in through the dorsal root ganglia from the entire spinal cord, from cervical all the way down to the sacrum. Also, some nerves are going to be encapsulated. So they might be encapsulated by these special receptor cells, right? And so we'll talk about that in a second as well. All right. Now, the way that these cells are partitioned is through embryological development. So these are going to be from during embryogenesis, where you have all that segmentation. Remember we talked about like the homeobox genes, right? The Hox genes, where you have segmentation of neurodevelopment during embryogenesis. This is also going to be something that creates these different dermatomes in your body. So the dermatomes, the specific definition is the territory innervated by each spinal nerve from a single dorsal root ganglia. So it's not necessarily the sections of your vertebrae, it's specifically from the dorsal root ganglia. And that can give you an approximate guess as to where, if you had nerve damage, where the damage was arising from, at what level. These are going to vary from person to person, so it's not like entirely precise. It gives you an estimate as to at which level of the spinal cord you might have damage, but they do overlap. Touch, pressure, vibration, right? You're going to see that being represented by these different dermatomes. And if you do get injury to the dorsal roots, you don't necessarily get complete loss of sensation because there is overlap between different ganglia, right? Not all ganglia are going to be responsible for one single dermatome. So here's your dermatomes, right? So remember, face, head, that's going to be associated with your trigeminal ganglia. Okay, whereas everything else, so like the posterior portion of your head all the way down to your feet, that's all going to be involving the different dorsal root ganglia from C1 all the way down to the sacrum. And so if you had deficits, for example, if you had like a spinal cord injury at the level of like T10, for example, you would see deficits going from the dermatome of T10 and below, right? And so that's one way you can figure out at what level you had the actual spinal cord injury. So here are your dorsal root ganglia. Again, dorsal root, that's going to be where you get sensory input, sensory afferents, and you have pseudo-unipolar cells. So I put different types of cell shapes over here. You got unipolar cell, we have the cell body on one end, and then you have the axon, which is myelinated all the way down to the axon terminal. Bipolar, you have the cell body in the middle, and you have two axons splitting off from each end. And this is the pseudo-unipolar, where you have the cell body, and it basically bifurcates to two different sides. And then a multipolar has extensive dendrites coming off of the soma, or the cell body, and then an axon coming off the opposite direction. So with the pseudo-unipolar cells, you're going to have a branch that's going to be associated with the PNS, so the peripheral nervous system, and then a branch that's going to be associated with the central nervous system. So you have peripheral nervous system afferents coming in, and then eventually you have those cells coming into contact with the central nervous system. Okay, and then that's going to be associated with, for example, like the spinal thalamic tract, right, going all the way up to the thalamus. There's a lot of diversity in these cell types. So there are going to be different sizes. There's going to be different types of conduction velocities, right, different stimuli that they're going to be responding to. So there's a lot of different cell types involved in these processes. So before we get into those cell types, let's talk about receptive fields. So we already mentioned the 2-point discrimination test. That would help figure out exactly how granular and detailed the receptive field is, or how not detailed the receptive field is. So receptive field is going to be an area that you come into contact with where you actually get action potential firing if you come in contact with that area. They, again, are going to be region specific. So like receptive fields on your fingertips and your hands are going to be different than the ones that you find on your trunk or your torso. They do overlap, and if you see overlapping of the receptive field, that increases the sensitivity of the receptive field. And the size of the receptive field, again, you can determine the size and the accuracy of the region that you're testing with the 2-point discrimination test. And again, different receptive fields are going to be associated with different cell types. So for example, Ruffini cell types are going to be not super discriminate in terms of its location, whereas Meissner's and Merkel cells are going to have much more of a distinct receptive field, especially Merkel cells. The receptive field in a Merkel cell is really, really precise compared to that you would find in even Pacinian or Ruffini type cells. And so here is a way to graph out in a histogram how those receptive fields operate. So in this graph, you have different regions that are going to be color-coded. So green is going to be these two regions that are really close together, and A, B, and C are going to be different overlapping receptive fields. Okay, so if you stimulate the green over here, you're going to see that the area B, which is like adjacent to all, it's involving the lateral ones, A's and C, as well as the receptive field in the middle, B. And so that's going to show a lot of accuracy when you're stimulating that one area. So you see that B is the area that's being perceived, right? And so that's the greater amount of sensitivity in that one region in B. And then A and C, not so much in terms of the actual like reception of the stimulation from the afferents. And then the red lines over here, that'd be like stimulating those regions right there, which lay within the fields of C, B, and A. So it's kind of equal distribution, right, as you start moving further away. And then as you move further away, even still, now you're basically stimulating A and C almost entirely, right? B in the middle is not being stimulated whatsoever in terms of like the actual sensory input. And so that would be where you would like take the caliper, right? So either you're using that little round tool for the 2-point discrimination test that has like different degrees of separation, or you could use a caliper, or you could just use, two toothpicks or two sharp objects to like, stimulate those regions on the skin. Notice that the tip of the finger, you don't have to have much distance between the calipers to be able to get a lot of, information, right? So you can actually see a greater amount of detail in the 2-point discrimination test compared to the wrist or other parts of the body where your calipers are going to have to be pretty spread out for you to actually begin to detect that it's two different points being stimulated. So long story short, finger is going to be a lot more sensitive than the wrist or other parts of your body. So here's another way to basically graph that out in a histogram. So this is the mean 2-point discrimination threshold, right? In other words, the distance between the calipers by which you would get actual stimulation, or not stimulation, but discrimination of those 2 points. So notice your fingers, your thumb, finger 1, these are really, really sensitive. So you don't have to have much distance between the two calipers. Whereas once you get up to the arm, the shoulder, forehead, right, even parts of your face is pretty sensitive, right? It's much more sensitive than other parts of your body, but not even nearly as sensitive as your hands, right? When you look at the picture of the homunculus, the face is pretty exaggerated, but the hands are especially exaggerated when you see the depictions of a homunculus. Hands are way more sensitive. Breast, back, belly, thigh. You have to have quite a bit of distance between the calipers to actually begin perceiving the changes in sensation. All right, so slow versus rapidly adapting afferents. With a slowly adapting afferent, it's going to continually fire as you have a stimulus in place. So you're stimulating the region, right? Whatever it might be, maybe electrode, maybe it's just touching the area, right? So that's going to fire continuously throughout the duration of the stimulus. And with the slow adapting fibers, one of the purposes of that is going to be for determining the size and shape of the stimulus. And then there's rapidly adapting afferents. So upon stimulus, you're going to see the rapidly adapting afferents fire, right? But then they kind of stop. Even throughout the duration of the stimulus, they stop. Until sometimes they'll fire again once the stimulus ends. So fire once at the beginning of the stimulus stops, and then there was my fire again. And this is going to give information about movement of the stimulus. So for example, clothing. So if you feel clothing on your body, right, And if you're moving around, that would be something that you might be able to detect with these rapidly adapting afferents. Yeah.
I don't know. To me, it sounds weird. I would think that slowly adapting would not mean that the stimulus would be continuous. Whereas rapid, you would think that it'd be consistent. I don't know. It's just.
Well, rapidly adapting because it just fires as soon as it gets a stimulus. And then if it stops, you might also see another firing of the neuron. Versus slowly adapting, it fires in more rapidity at first, but then it keeps on firing, right? It doesn't stop until the stimulus stops.
So like if you're working out, it's like slowly adapting.
Yeah. So this is like the example from the textbook, carrying a heavy object. Yeah. Can't really think of any other good examples off the top of my head, but carrying a heavy object, you get slow adaptation versus something just like feeling touch from your clothes. It's going to be rapidly adapting. And those are the examples. I can't really think of any other good examples to help you with that. But so again, the slowly adapting stimulus is going to continue to fire as a response to the stimulus. And then the rapidly adapting receptors basically just fire at the onset and sometimes when the stimulus ends. This can also help you determine the qualities of the stimulus, like static, like, for example, carrying a heavy object, right? It's going to be static versus something that's more dynamic. Free nerve endings. Again, free nerve endings are what are responsible for pain detection. And so as soon as you get maybe like a puncture wound or a cut, those free nerve endings are going to start firing like crazy, right? Providing pain input. Again, another reason why if you cut your finger, put it in your mouth, that helps to alleviate the pain a little bit. And here are some of the other cells that are going to be important for sensation. So we're going to go into all these in more detail. Merkel cells, Meissner's, Pacinian, and Ruffini corpuscles. So for Merkel cells, these are going to be associated with light touch. And on top of that, these have a very high acuity in terms of reception. So these are very, very sensitive. So if you notice on this image over here, where it has like the different cell types of the fingers, right? Merkel cells, very tiny little area, right? So the sensory field is going to be much smaller, meaning that it's going to be much more detailed. Versus Ruffini, those are going to be a more kind of like diffuse, not super defined receptive field, so not as specific. So Merkel cell, long story short, the spatial acuity is really high. They're also slowly adapting, lots of myelination. In terms of myelination, you'll see some of these cell types are myelinated, some are not myelinated. These ones happen to be myelinated. Meissner's cells, this is going to be associated with fine touch, discriminative touch, as well as vibration, so low frequency vibration specifically. You'll find these close to the skin. So in this diagram over here, you can see the location of all these. So you got Merkel cells, Meissner corpuscle. The deepest ones are going to be Pacinian, and Ruffini are kind of like mid in terms of depth, right? Mid-range. So the Pacinian, that's going to be gross pressure. That's the deepest one, is the Pacinian. And Pacinian is going to be due to like more intense pressure changes. Also vibration. So you have two of these that are going to be dealing with vibration. Meissner's and Pacinian both deal with vibration. Meissner, low frequency vibration, and then Pacinian is going to be just like vibration in general. But long story short with this is that it's deep inside the dermis. Then the Ruffini cells, those are going to be associated with stretching. They're going to be associated with movement. They're going to be associated with, like, for example, finger position. And those are also pretty deep, but again, not as deep as the Pacinian. Pacinian are the deepest. Ruffini are going to be the second deepest. Meissner's a little bit above that. And then Merkel's are going to be really close to the epidermis. So here is an example of a cross-section of glabrous skin. This is going to be on the fingertip. And you'll see all of these cell types are going to be present. Okay, you're going to see all these in your fingertips. Hairy skin is a little bit different. You get different types of cells. You get like these circumferential cell endings that are associated with the hairs. There's also these longitudinal endings, right? So hair is going to involve different kind of mechanisms when it comes to like touch perception. We're not going to go into those in too much detail, but make sure you're familiar with your Meissners, Merkels, Pacinian, and Ruffini. They're all specialized, so they all do something different, right? They're all going to be receiving either information that's associated with like fine touch, vibration, stretching, right? So these are all going to be specialized. So there's a lot of variability in these cells. And here's an example of a study. This was featured in your textbook as well, where there's somebody who's reading Braille. There's going to be different cell types that are associated with different types of sensation. So for the Merkel cell, helps with detail, right? Remember with Merkel cell, very tiny receptive field, so it's extremely detailed. Meissner cell, you're going to feel more of like the coarse type sensation. Pacinian and Ruffini are going to be the deeper ones, right? And they can allow for movement perception as well as position perception. Hair, these are your hairy cells, right? You're going to find Merkel cells, right? You're going to find these longitudinal cells as well that are associated with like the movement of the hair, whether it's like stroking or wafting of air across the skin. They're rapidly adapting, right? So in other words, they experience the sensation from the stimulus, they stop firing until the stimulus ends, and then they might fire again, basically indicating that the stimulus is end. There's different subcategories of cells, A beta, A delta, and C fibers. We're going to talk about A delta and C fibers when we get into pain sensation, because those are the ones that are especially associated with pain. A beta, mostly associated with just like touch. So mostly like innocuous, benign type of sensation. But we're going to talk more about those in the next section. And the circumferential, that's going to be around the circumference of a hair follicle, and that's going to be also associated with stroking sensation. Now, in terms of the receptors for touch, we talked about these before, but the piezo family receptors are going to be involved in mechanical stimulation. Now, if you recall from when we discussed receptor types before, the piezo family receptor types have 3 subunits, and they kind of fan out. So think about them as being like kind of flat, almost like plate-like. Right? It's like a dinner plate. Right? But then as soon as you start manipulating the area, the skin, it's a mechanoreceptor, then you start changing the conformation of the actual receptor when you're like applying pressure, for example, to the area. And that is basically what opens up the channel. And it's an ion channel. As soon as it opens up, all of a sudden ions start rushing in. And that has to do with basically mechanical manipulation, mechanical stimulation of those cells. And in the example here, it's sodium ions coming in, right? So sodium ions rushing into the cell. And then once you get enough stimulation, that's when you actually get like an action potential where the cell actually fires, once it hits the threshold. But if it's just weak stimulation, you're not going to get any sort of effect, right? It has to be a stronger type of stimulus. So these are going to be your alpha beta type of cells, right? So remember, alpha beta is going to be mostly associated with touch. Alpha delta is pain. Okay, so make sure you know the distinction between those two. So again, this has to do with manipulating the texture and the doing like deformation, right? You're manipulating the actual receptors in the cell to allow ions to come in. And then eventually you get depolarization. Now, in terms of proprioception, that's going to be involving a lot of different cell types within your skeletal muscle. And so you have different types of axons, both efferents and afferents, that are associated with your muscle. So you have your alpha motor unit neurons. Those are going to be associated with depolarizing the skeletal muscle at the motor end plate to allow for contraction. Contraction is predominantly going to be associated with extrafusal muscle fibers. Okay, so whenever you contract the muscle, you're thinking extrafusal muscle depolarization. The intrafusal muscle fibers are going to be associated with sensory input. And so you're going to have afferents coming in from your intrafusal muscles, and those are going to allow for basically a balance. They want to detect any sort of like abnormalities in muscle contraction. So it's really important to allow for like coordination of muscle contraction to make sure that muscle contraction is working properly. And so these are all part of your muscle spindles, including your intrafusal muscle fibers. Sorry, your extrafusal muscle fibers. Those are all falling under the category of muscle spindles. But the ones that are really important for contraction in particular will be extrafusal muscle fibers. Intrafusal is sensory for the most part. Yeah.
Is it just for contraction purposes?
Yeah. Yeah. Then another thing that's important as well is your Golgi tendon organ. So I'm sure you guys remember some of these from when you took A&P, for those of you who took A&P. I know some of you probably didn't take A&P. You didn't take A&P, right? No. But most of you guys took A&P, right? So you guys went over this in part 1 of A&P. So Golgi tendon organ. What does the Golgi tendon organ do? Do you guys remember? Have you guys ever picked something up that was too heavy and you dropped it immediately? Yeah, that's basically what's going on here. So the Golgi organ tendon is important for protecting the muscle from excessive strain, excessive stress. So these are going to involve low threshold mechanoreceptors. And if you exceed the threshold for these muscles, then you're going to see immediate relaxation of the muscle. And that's a protective mechanism. So this protects the integrity of the muscle. So these are going to detect changes in muscle tension, right? And again, if it goes beyond a certain threshold, then you'll see immediate relaxation. So that's part of the Golgi tendon organ reflex. You have different types of mechanoreceptors in joints as well. I'm not going to really hold you guys accountable for all of these, but you have 3 different types, type 1, 2, and 3. They're going to be involving slow and rapidly adapting afferents, but I'm not necessarily going to hold you guys accountable for those. But make sure you know your Golgi tendon organs. Make sure you know your extrafusal versus intrafusal muscle fibers. And these are all going to be associated with proprioception as well. And recall, proprioception is how you are able to determine where your appendages are in 3-dimensional space. Muscle spindles, again, they're going to have extrafusal fibers. They're also going to have intrafusal fibers. Then you're going to also have the Golgi tendon organ being associated with this as well. So these are all going to be playing a role in proprioception of your skeletal muscle. So any questions on any of that? So know your major fibers, okay? Know what those all do in terms of either a protective mechanism like the Golgi tendon organ, whether it's just mainly for the function of contraction, extrafusal, or afferent detection of how muscles are operating when it comes to intrafusal muscle fibers. Again, dorsal horn, that's associated with input, sensory inputs. All of your afferents go through your dorsal horn. There's going to be interneurons associated with your spinal column as you get afferents coming in. Some are going to be excitatory, some are going to be inhibitory. And then you have neurons that are projecting all the way up to the brain, right? They're going to be reaching all the way to the level of the thalamus. And from the thalamus, they're going to go to the somatosensory cortex. So we'll talk about that in a little bit. You also have proprioceptive inputs through the ventral horn, and those are going to be associated with the motor nuclei in the skeletal muscle. So now let's get into the different ascending pathways that we're going to be discussing. So dorsal column medial lemniscus pattern. That's going to be a allowing for sensory input from the entire body, as well as the posterior portion of the head. Remember your dermatomes, right? That's going to involve the dorsal root ganglia going all the way from your cervical spine all the way down to the sacrum, except for the trigeminal ganglia. So the trigeminal ganglia would be for the face, but the posterior portion of the head is also going to be part of the cervical ganglia. Again, trigeminal portion is going to be for the face. Let's play a little video that talks about. Oops. Let me turn this up. All right, hold on a second.
Welcome to 2 Minute Neuroscience, where I specifically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss touch and the dorsal column's medial lemniscus pathway. Somatosensation involves sensations from the body. When these sensations come from the external environment, they are picked up by receptors in the skin called cutaneous receptors. There are several different types of cutaneous receptors, each designed to respond to specific types of touch sensations, like pressure, pain, or vibration. In this video, I'll discuss signals that involve information about fine touch and vibration. I'll cover pain in a separate video. The main pathway that carries information about touch to the brain is the dorsal column's medial lemniscus, also called the posterior column's medial lemniscus. This pathway also carries information about proprioception, or the position of the body in space. These signals come from proprioceptors in the muscles and joints. Fibers in the dorsal column's medial lemniscus leave cutaneous receptors or proprioceptors and enter the spinal cord via the dorsal roots. They travel up the spinal cord to the medulla in one of two fiber bundles within the dorsal columns, the fasciculus gracilis, which carries information from the lower half of the body, or the fasciculus cuneatus, which carries information from the upper limbs and torso. In the medulla, the fasciculus gracilis and fasciculus cuneatus synapse on the next neurons in the pathway in areas called the nucleus gracilis and nucleus cuneatus, respectively. It is from these nuclei that the second part of the pathway arises, a fiber bundle called the medial lemniscus. The medial lemniscus leaves the dorsal column nuclei and quickly decussates or crosses over to the other side of the brain before traveling up to the thalamus, where it synapses in a part of the thalamus called the ventral posterior lateral nucleus, or VPL. A third part of the pathway arises from the thalamus and travels up to an area of the cortex called the postcentral gyrus, which contains the main sensory area for touch in the brain, the somatosensory cortex. Specific parts of the somatosensory cortex receive signals from specific parts of the body, an arrangement that is known as somatotopic. Information about the nature and location of the sensation are integrated in the somatosensory cortex, where the conscious perception of the sensation begins.
Good stuff. I love that guy. He does a great job explaining stuff. So again, as he mentioned in that video, you got the gracile nucleus, or part of the gracilis pathway, and then you have the cuneatus pathway as well. So gracile or gracilis, it's lower extremity, so maybe think like a graceful dancer, perhaps. Maybe that might help. Cuneatus is going to be upper portion of the body. Cool. So you got your gracile nucleus, gracile tract, lower part of the body. It's going to be coursing medially. So if you look at the cross section of the spinal cord at the dorsal columns, you're going to see like 4 little bumps, right? And they're in pairs, right? There's going to be one that's more medial, and that's going to be the gracile tract. And then the one that's a little bit more lateral, that's going to be the cuneatus tract. Again, cuneatus is going to be upper body, so neck, trunk, and the upper extremities as well. Now, you're going to have second order neurons coming from the dorsal column. So there's like 3 major types of neurons here. You got the ones that are part of the peripheral nervous system, right? So you're going to see peripheral inputs coming from your afferents. And then once they come in contact with the spinal cord, then you start seeing your second order neurons. And the second order neurons are the ones that make them way all the way up to the thalamus. And so you're going to have internal arcuate fibers. That's going to be associated with the dorsal column, and then the medial lemniscus, and that's going to be the one that basically decussates at the midline, so crosses over. And then those are all going to make their way all the way to the thalamus. The specific region of the thalamus is the ventral posterior lateral thalamus, VPL. Okay, so ventral posterior. So ventral, It's going to be on the bottom of the thalamus, posterior towards the back, and lateral. So it's going to be on the bottom and the back on the lateral sides. So here's your trigeminal nerve. Again, that's going to be associated with your face, right? So head and face. It's going to be associated with the trigeminal ganglia. And you have your trigeminal ganglia here. It's going to synapse with the nucleus of the trigeminal complex. So you have the principal nucleus, and then you also have a spinal nucleus as well. Not super high yield. Just know your trigeminal ganglia and how that's completely separated from all the other ganglia along the dorsal column. So these are all going to be associated with different types of sensory input, like pain, touch, temperature, etc. And so still part of your trigeminal pathway, you're going to have your second order neurons, right? So you have basically your peripheral neurons, your cranial nerve 5, and then you're going to have your second order neurons coming off of the trigeminal ganglia to the trigeminal lemniscus, and that makes its way all the way up to the thalamus, ventral posterior medial thalamus, whereas the other ones were lateral, right? So the medial lemniscus fibers, for example, they're going to make their way up to the ventral posterior lateral, versus trigeminal is ventral posterior medial thalamus. So, proprioception, again, we're going to talk about proprioception with the lower parts of the body. This is going to be involving afferents that are associated with Clark's nucleus. So that's going to be this region right here within the spinal cord. And then it's going to be associated with the dorsal spinal cerebellar tract. So with proprioception, the cerebellum, remember the cerebellum is going to be really important in fine-tuning movement. It's going to be associated with posture, things like that. And so when it comes to proprioception, the cerebellum is going to be really important in that whole process. And so with these fibers, they're going to be making their way up to the cerebellum. So that's part of the dorsal spinal cerebellar tract. So that's lower body. When you get to the upper portion of the body, you're going to see dorsal column nuclei, and that's going to be this pathway right here. That's going to be associated with the external cuneate nucleus. You also have the mesencephalic trigeminal nucleus. That's going to be associated with the face. So that's going to be proprioception for the face. All right, so these are the thalamic nuclei that are associated with sensation, right? We talked about the ventral posterior medial nucleus associated with the trigeminal system, aka the face, so facial sensation. And then for the rest of the body, it's going to be the ventral posterior lateral nucleus. So medial, face, lateral, rest of the body. Let's watch a little video on that.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the thalamus. There is a thalamus in each cerebral hemisphere just above the brainstem. The thalamus is often described as a relay station because a great deal of information that precedes the cerebral cortex first stops in the thalamus before being sent on to its destination. The thalamus is subdivided into a number of nuclei that possess functional specializations for dealing with particular types of information. For example, all sensory information except olfaction travels directly from sensory receptors to a nucleus in the thalamus specialized for dealing with that type of sensory data. Then the information is sent from the thalamus to the appropriate area of the cortex where it is further processed. As many as 50 distinct nuclei have been identified in the thalamus, but I will discuss just a few of the better known nuclei in this video. Each of these nuclei has multiple functions. I will only mention one or two of the best known functions here. At the anterior of the thalamus is a nucleus called the anterior nucleus. It is extensively connected to the hippocampus and is thought to be involved in memory. The dorsomedial nucleus is thought to be involved in emotional behavior and memory. The ventral anterior nucleus and ventrolateral nucleus are thought to be involved in motor functions. The ventral posterior lateral nucleus, or VPL, and the ventral posterior medial nucleus, or VPM, both act as relay nuclei for sending somatosensory information to the somatosensory cortex. The lateral posterior nucleus is thought to be involved with integrating sensory input and associating it with cognitive functions. The pulvinar nucleus is a large nucleus that is involved in processing visual stimuli. The medial geniculate nucleus and the lateral geniculate nucleus serve as important relays for auditory and visual information, respectively. The reticular nucleus forms a sheet that makes up the outer covering of the thalamus. It influences the activity of other nuclei within the thalamus. There are also a number of nuclei not visible in this image, such as the centromedian nucleus, which is thought to be involved in attention and arousal.
Okay, so the thalamus, super complex. You obviously don't need to know every single detail about the thalamus, but if you are comfortable with the ventral posterior medial and the ventral posterior lateral nuclei and which regions of the body they're getting information from, then you should be good in that regard. So again, we're revisiting the homunculus. So here's your homunculus. As I mentioned moments ago, really big hands, right? Even some of the facial features are going to be a little exaggerated as well, especially the lips, right? But the homunculus, you see this disproportionate sensitivity represented from different parts of the body. And then you have different areas of the somatosensory cortex. So you have these different subregions. You have like A3A, A1, A2. I'm not going to hold you guys responsible for all of these, but we'll refer to a couple of these different regions in a moment. We already talked about that. So here's your cortico-cortical connections. So cortico-cortical, meaning they have one region of the cortex basically being associated and communicating with other regions of the cortex as well. So there's going to be a hierarchy of processing when it comes to all these different regions. So S1 is going to be part of your primary somatosensory cortex. So if you look at Brodmann's areas are going to be numerical values associated with all the different regions of the brain. And so when we talk about Brodmann's area S1, we're talking about the post-central gyrus, right? The somatosensory cortex. Primary somatosensory cortex is going to be labeled as S1. And the primary somatosensory cortex is associated with things like texture, orientation, direction, speed. There's lots of different functions that are going to be processed by the primary somatosensory cortex. And if you were to do like a breakdown of all these different pathways, which I'm not going to hold you guys responsible for, but these are all the different pathways associated with the somatosensation. So you have inputs coming in through the thalamus, and then it gets relayed from the thalamus to the higher cortical areas, right? Because remember, the thalamus is basically just a relay point, right? So it's going to be relaying information to different regions. So 3A, 3B, 1 and 2, these are all part of the somatosensory cortex, right? And then that information can get sent even further to the limbic system, to the amygdala as well as the hippocampus, right? Amygdala, especially if it's like associated with fear detection, right? Threat perception, because you respond to environmental stressors, and that's going to be processed by the amygdala ultimately. So it can actually help you determine what kind of reaction you want to engage in as a response to that sensory input. Hippocampus, right? You want to basically put things that are like sensory input, you want to consolidate that into memory, right? So you learn different types of sensation. You learn, for example, what's a pleasant stimulus, what's a unpleasant, noxious stimulus, right? So this all has to be involving the amygdala and the hippocampus at some point. And then also you're going to be sending information from the sensory input to the frontal lobe, right? So that you can get some sort of reaction to that information, right? So you can see what type of motor reaction you're going to want to engage in. So there's another breakdown of all those different regions and Brodmann's areas. I'm not going to hold you guys responsible for all these different little subregions, but be familiar with, whenever you hear what a Brodmann's area is, it's just basically a numerical value associated with different areas on the cortex. In terms of cortical real estate, we've already kind of touched on this before, but you're going to have areas of your cortex that are going to be responsible for a specific regions of sensory input. So a good example is with rats and mice, right? They got their whiskers on their face. Each one of those whiskers is going to correspond to a different region in their somatosensory cortex, right? So if you stimulate a single whisker, that one area is going to light up. If you were to do like a little fMRI on this rat, you'd be able to see that one tiny region lighting up. So here's cortico communication, again, from, you get sensory input, then you have that information being processed in one region of the cortex, and then that information gets relayed to a different region of the cortex, so you can figure out exactly what kind of response you want to engage in, secondary to that stimulus. Neurons in one cortical area communicate by exciting their target neurons in other cortical areas. And as you can tell from this study over here from Gambarini and colleagues, 2021, that's some of these pathways are extremely complex, right? They involve many different cortical regions, many different subcortical regions in the brain, right? So these pathways can be very complicated. Now, we discussed neuroplasticity before, especially experience-dependent neuroplasticity, right? This would be an example of that, right? Experience-dependent. The more you stimulate a region, the more that area is going to be represented in the brain, right? And then the opposite is true. If you stop stimulating a part of your body, that area in the brain atrophies. So that can happen to a person who gets an amputation. Right? We're going to talk about phantom limb pain in a little bit, but that's a different process. But if you don't use it, you lose it, right? So if you lose a finger, for example, that region in the brain that corresponds to that finger is going to eventually atrophy, right? And then the other regions corresponding to the remaining fingers are going to become like more represented on the cortex. And so in this example, they removed one of the fingers. I think it's the third digit in this case. The fourth, fifth digit, second digit, all those would increase in their representation on the cerebral cortex. So this was a study done on a monkey brain, owl monkey. And the changes can happen within months, right? This is like 2 months after amputation of that finger. Within 2 months, all of a sudden, that region in the cortex no longer had much representation. So pretty cool stuff. And then again, the opposite is true, right? So if you hyperstimulate an area with sensory input, that area is going to start getting more and more representation of the cortex. So for this case, they were hyperstimulating, I think for this case, it was like the third and fourth digit, actually, as well as the second digit. And those areas eventually had more of a representation of the cortex compared to the fifth and first digit. Yeah, so in this case, they made they subjected monkeys to different tasks that required more movement of the second, third, and fourth digit. And then over time, those areas basically increased in their representation. Now, with hepatic touch, this is going to be involving a combination of exploratory movement plus tactile feedback, right? I mean, we have hepatic touch features on our smartphones, right? Whenever you like touch your phone, right, there's going to be hepatic touch features on that. So some of the mechanisms that come from neuroscience have also been used in some of our technology nowadays as well. All right, now if you have dysfunction of the somatosensory system, one type of dysfunction is going to be Friedreich's ataxia. What's undergirding Friedreich's ataxia? It's going to be a genetic mutation of the frataxin gene. And so you're getting these trinucleotide repeats. Trinucleotide repeats can also be seen in other genetic diseases as well. We're going to talk about Huntington's later on, which also involves trinucleotide repeats. And over time, with those trinucleotide repeats, you're going to see dysfunction at the level of the mitochondria. And with mitochondrial dysfunction, with this case in particular, you're going to see inability to coordinate, movement, posture is going to be affected, walking is going to be affected, manipulation of objects is going to be affected. So let's watch a couple different videos. they're sad, right? These types of diseases are really, really sad conditions, right? So hopefully nobody cries. If I cry, I apologize. It's because I have allergies.
Ataxia is a disorder where there's impaired mitochondrial function that results in damage to various organ systems. In particular, the nervous system gets damaged, which causes ataxia, where the muscles cannot be moved in a coordinated way. The disorder also affects other organs like the heart and pancreas. The disease gets its name from the German physician Nikolaus Friedrich, who first described the disease over 150 years ago. So normally on chromosome 9, there's a gene called the FXN gene that encodes a mitochondrial protein called frataxin. The normal amount of frataxin varies by tissue, with some tissues like the nervous system, pancreas, and heart containing lots of it. Frataxin helps put together cofactors called iron-sulfur clusters. It is a combination of iron and sulfur that form part of enzymes with many functions, such as electron transfer, which is a key part of mitochondrial ATP production. Friedreich's ataxia is caused by a mutation in the FXN gene where there's an abnormal repetition of a GAA sequence within that gene. This is called a triplet repeat, or trinucleotide repeat, which means that a group of 3 DNA nucleotides is repeated multiple times in a row. In this case, guanine, adenine, and adenine. Normally, the GAA sequence is repeated 7 to 34 times within the FXN gene. But in Friedreich's ataxia, there is a repeat expansion where there are 100 to 1,700 times as many copies, with most individuals having repeats ranging from 600 to 1,200 times. Now, Friedreich's ataxia is inherited as an autosomal recessive condition. It's passed on by parents who are carriers because they have one expanded FXN gene and one normal FXN gene, but they don't have any symptoms of Friedreich's ataxia. They end up passing on their expanded FXN genes to their kid. Inheriting both copies of the FXN gene with an expanded GAA repeat is the most common way to get Friedreich's ataxia. The repeat expansion causes gene silencing, which is when the FXN gene is not transcribed normally, and very little frataxin protein is made. With low levels of frataxin, the mitochondria are unable to efficiently incorporate iron into iron-sulfur clusters, and as a result, there is lower mitochondrial ATP production. So there's less energy available for the cell. Furthermore, iron accumulates inside the mitochondria, which reacts with oxygen to create unstable oxygen radicals. Over time, these free radicals damage DNA and proteins in the cells in a process called oxidative damage. Then, this energy deficiency and oxidative damage result in dysfunction and death of the cells that are highly dependent on mitochondrial function, such as neurons, cardiomyocytes, and pancreatic beta cells. The loss of neurons leads to ataxia. In the heart, there is abnormal thickening of the ventricles, a condition called hypertrophic cardiomyopathy, which is the most common cause of death in people with Friedreich's ataxia. In the pancreas, beta cells die off, leading to decreased insulin production and diabetes mellitus. In individuals with Friedreich's ataxia, the ataxia usually begins in childhood, which causes them to have difficulty in walking. They might have some muscle weakness and loss of proprioception, which is the sense of your position in space relative to other objects. Usually, it affects the legs and then moves up to the torso. A generalized feeling of fatigue is also common. Eventually, most people with Friedreich's ataxia need a wheelchair to get around. In later stages, they may develop complications of cardiomyopathy, such as heart failure, and some even end up losing their vision and hearing. The diagnosis of Friedreich's ataxia is established by genetic testing. This test is used to identify the expanded GAA repeat in both copies of the FXN gene, the most common cause of Friedreich's ataxia. Additional tests may be performed to help determine the extent of the disease, such as an MRI of the brain and spinal cord, and echocardiography to measure the heart wall thickness. There is no treatment that can slow or halt the progression of Friedreich's ataxia, but symptoms can be managed. For example, physical therapy can help maintain muscle function. Alright, as a quick recap, Friedreich's ataxia is caused by a repeat expansion in the FXN gene on chromosome 9. It is normal to have less than 34 GAA repeats, but it is pathogenic if there are over 100 repeats. Friedreich's ataxia affects the nervous system, pancreas, and heart due to defective mitochondrial function and oxidative damage. Ataxia, diabetes, and hypertrophic cardiomyopathy result.
All right, that wasn't the sad video I was talking about. So,
I was waiting for this.
yeah, well, it's gonna happen right now. There's a bunch of these, by the way. If you want to like go down like a really depressing rabbit hole, just watch all the videos of people with Friedreich's ataxia.
My name is Sam Bridgman. I'm 28 years old, and I was diagnosed with FA at 15. Tried to get symptoms probably when I was around 12. I would always play baseball with my dad, and he would hit it to me, and I would have trouble tracking the baseball. So instead of me getting better in sports, I was getting worse. I would trip and fall on the ground. My parents would ask me what happened, and I'd be like, yeah, the ground jumped up and tripped my foot or something. I think my parents thought something was wrong, but I didn't really think anything of it. And it took about 3 years to eventually go to the neurologist and do a blood test for FA. I mean, high school is a tough time for everyone. And when you add something like an FA diagnosis, that makes it even harder. I did not tell for probably a good year and a half. Just keep living my life like I did not have FA, pretty much. I would say to not wait to tell your friends about FA. It's always good to have friends and family that are by your side. The friends I told are my best friends today. My tattoo says "Seek a miracle," and impossible is nothing. With baseball stitching in between, my parents and my brother all have the same tattoo. Seek a miracle because FA doesn't have a cure. We're seeking a miracle every day. And then impossible is nothing, and it's kind of always stuck with me, and that is my life motto that I live by. Life is not over. Keep moving forward every single second or every single day.
All right. I know, right? It's very profound, very powerful stuff. But there's a lot of stories like that. Lots and lots of stories like that. Just wait until we get to Huntington's. I'll show you some videos that are real tear jerkers. Other things that could affect the somatosensory function. Parkinson's disease, right? The hallmark of Parkinson's is going to be alpha-synuclein accumulation. These are going to be accumulated in the form of Lewy bodies. And what happens is you get destruction of the basal ganglia, right? The substantia nigra, pars compacta within the midbrain. And then you're not going to get dopamine production. And then you're not going to get the basal ganglia functioning properly, which we're going to go into excruciating detail in a couple weeks. And then rigidity, right? That's one of the big things. Rigidity, poor balance. They also get like a really bad resting tremor, coordination issues. And then there's also autism spectrum disorder, or ASD. They're going to have a lot of impairments in touch, right? So impairments in non-discriminative affective touch, as well as sensitivity to light touch. When I was when I was your guys' age, when I was in my undergrad, I worked for about 1.5 years with an autistic guy. He was actually a family friend of ours. I was working for this local company that they work with like special needs, mostly autistic patients. And he was like 2 years younger than me. So I was about 21 at the time. He was about 19. And I worked with him for 1.5 years. and he spoke, within a year and a half, he spoke 4 words to me total. So he had really, really severe autism. One thing that you see with autistic patients, especially if it's like really severe, is that they have a really hard time with just like communication in general. Like even parts within their brain just don't really communicate properly. And a lot of it's still not really well understood when it comes to autism. No. Oh yeah, absolutely. That's what I was getting at. So overly sensitive to light touch. They're also overly sensitive to a lot of things. They're overly sensitive to sound. They don't like breaking routines. They really like a routine. So they're very, very, like the very severe cases of autism are very difficult to manage just because it's like they are completely 100% dependent on others. But there are cases of autism that are not as severe, right? That's why it's a spectrum, right? Autistic spectrum disorder. Some people are highly functioning, and I'm talking high IQ, and they're really able to function properly. Asperger's would fall under that category. A lot of people that work in tech in Silicon Valley, a lot of these people are Aspies. They are high performers. But the thing with like Asperger's is that they don't really pick up on social cues very well, right? So sometimes they might be perceived as rude or kind of like aloof, right? They don't really have like good communication skills. I was listening to a story about this lady. She was a surgeon and she had Asperger's, and she was really like kind of like, she didn't have like much in terms of social skills, right? So people perceived her as being very, very rude. They actually did a deep brain stimulation on her, targeting a certain region of brain. I don't know which region was it specifically, but when they stimulated that region of the brain, it actually made her more empathetic during the deep brain stimulation. So she was actually able to like finally be nice to people, but it was only while the deep brain stimulation was taking place, right? As soon as they turned it off, she went back to normal. And so she was just always kind of like the way she was, right? Just kind of very difficult personality, right? A little brash, maybe sometimes even too honest, right? That's one of the things that happens with people with autism. Yeah.
So it's like their thought process is to be nice, but then it comes out completely different, or do they think about being rude?
It really depends on who it is, right? Everyone's different, right? But the people who have really bad like personal, like interpersonal skills, right? They don't mean to be mean-spirited or like blunt or rude. That's just part of who they are, right? They don't have like a very good sense of empathy, right? So they have a hard time empathizing with other people. But again, like I said, it's a spectrum, so everybody's gonna be different. So you have autism, you have Asperger's. There's also pervasive developmental disorder. Like I said, some of these people can speak really well. They can have really high IQs, but in terms of their social skills are really not that great. And then if you're in a really severe case of autism, where you have like severe learning disability, your activities of daily living, you need like help with basically everything. Those individuals need like full-time care. So full-time caregivers. That's a long video. I'm not going to play the whole video. You guys can watch that video on your own time if you'd like.
What's pervasive developmental?
They just don't develop like across the board. It's like language development, like so speech, other types of communication skills, their learning abilities, right? Whether it's like reading, math, just basic knowledge, right? So if you have pervasive developmental disorder, it's pervasive meaning that it's like global, like all of your abilities are going to be lacking. Again, social interactions are going to be very poor with these patients. They have reduced interest in people. They don't really like to make eye contact. This dude that I worked with, very like skittish in terms of eye contact. Like you try to like talk to him, like, hey, what's up? He like looks away, he walks away. Right? If you say hi, he'll like literally turn around and walk away from you. Right? So it's just how they are. Delayed speech or no speech. Like I said, this guy, 1 year and a half, 4 words total. And it was amazing. I was like, oh my God, he spoke. Like, what did you say? That's cool. Like, I thought he was going to get better. But no. And, you know, like I say, he's a family friend. He's actually, his mom is Brazilian. My mom and his mom are like really close friends. And so she went over there for some holiday not too long ago, and she spent time with him. She saw him, and she said he's still exactly the same way. He's like 2 years younger than me. I'm 38. He's 36, and he's exactly the same as he was when he was younger. I am. I'm 38. I'm old. I'm an old man. Oh, thank you. I appreciate that. All right, guys. You're not gonna get extra credit for the compliments. Although I will take it. I appreciate it. They also might repeat words and expressions. If you see a young individual, I'm talking like toddler age, 2, 3, maybe even up to 4 years old. If you start seeing them doing like repetitive movements, like rocking back and forth and stuff like that, red flag. Like probably want to get some sort of like psychiatric or neurological evaluation on that kid, because he's probably has autism of some sort. Unusual interests, rituals, right? There was this kid that I worked with who had Asperger's at the first psychiatric ward that I was working at. And this kid was wild. He had an encyclopedic knowledge of movies. He knew like the names of the movies, the actors in the movies, the year the movie came out. I'm not talking about recent movies. I'm talking about movies dating back to like the 60s and 70s and 80s. And it was weird because like that's like a really weird thing to be like an expert in. And he was like a kid. He was like 14 or 15 years old. And like he had a heart, like he didn't talk about anything else. Like you couldn't talk to him about anything else. He would only talk about that. And so they have these like really weird interests. Like they really focus in on one thing only, and that's it. It's pretty amazing stuff. Again, poor eye contact, difficulty with expression, difficulty following directions. They do get upset if you change their routines. They overreact a lot. They really overreact to things that don't usually, bother people. They can be impatient. They often prefer to be alone. This kid I worked with, he would be in the shower. I would get there. His mom was like, oh, he's in the shower. I'm like, okay. He would be in the shower for like hours. I'm talking like 2 hours. Yeah, with the water running. I would like knock on the door and be like, are you okay? I would like kind of hear some rumbling in there. Like, okay, I guess he's all right. So anyways, not all people with autism are going to act the same. Each person has a unique strength and challenge. Each person displays feelings differently. They're very honest. So if you say, like, do I look fat in this dress or do I look fat in this outfit? They're going to be honest with you if you look fat. Difficulty recognizing social cues, right? So there are savants out there, right? There's been some famous, like, pianists that are just like incredible piano players, but they're just like totally autistic. Right, but not all people with autism are going to be savants. That's like a very small sect. There's this one guy I was watching a video on. He's autistic. He can listen to any note on the piano and recreate it. Even like horrible. Like if you just go bam and just like hit a bunch of crappy notes, he will hear every individual note and recreate that just by listening to it. Insane. Like insane ability. Like I fancy myself a pretty decent musician. Like there's no way in hell I could do that. Yeah, and that's what he does. He plays by ear. He's a magnificent musician. Like his abilities were incredible.
Have you seen The Good Doctor?
I have not. No, The Good Doctor. The Good Doctor. I'll put it on my list. Yeah, I'll definitely put that on my list. So I'm not going to go over this quizzes today. We're going to do these on Thursday, so I'll have time to finish those up. He says The Good Doctor.
Yeah.
I've heard of it. I've definitely. I've heard of it.
Is Sheldon Cooper like an example of Asperger's?
Who's Sheldon Cooper?
That's the dude from Big Bang Theory.
Oh, I never really watched that show, so I don't know. I think there is somebody. I think there is somebody who's got like Asperger's in there, if I remember correctly. I think somebody mentioned that in one of my classes. Yeah.
It seems like a lot of what you were saying.
Yeah. Anyways. Now, let's switch gears. Now let's talk about pain and temperature. So we're going to close out with this today. Hopefully we'll break through to the end, all the way through there. Speaking of breaking through, breaking down the gates of hell, this is a painting by one of my favorite painters, Hieronymus Bosch. I highly recommend looking at his art. Like his art is so schizophrenic and he's from the Renaissance period, and it's amazing. Like some of these panels are actually not that big, but like the detail and like the little tiny people. Like I bought a book that had like it goes into like little details of like each of the portraits, and it's a really big book, and like you go and like just incredible detail. But then all the characters are just really wild. Like there's this one's like the Garden of Earthly Delights, and it's like a bunch of people just like pursuing like hedonistic pleasure, and then he has depictions of hell, and it's like pretty gruesome, like people getting all sorts of like torture and stuff. It's awesome art, but amazing. So anyways, I thought it was suiting for Pain and Temperature. Ooh, I want you to watch this since we're watching Pain and Temperature. This dude is absolutely insane, and he goes around getting himself stung by different types of animals. And so the tarantula hawk, which is native to the Southwest, I'm sure some of you guys have seen these around. Yeah, they look like bats from hell, right? They're like some of the most terrifying things. The tarantula hawks, so they lay their eggs in tarantulas, and they parasitize the tarantula. The eggs hatch, then the larvae feed on the tarantula, and the tarantula die. The tarantulas never survive that. And they have the second most painful sting of any creature out there.
Now, look how much my hand is shaking. Hold on.
This guy's such an idiot.
Okay.
I don't know.
I got a really good hold.
Wow, look at that abdomen going.
I got a really good hold on her. It took me a minute to get the right hold, but the right hold is what is important. Now, I do have the glass capsule right here, so I'm hoping that I have the wherewithal after the sting to quickly pick up the glass enclosure and put it back over top of the wasp so it doesn't escape, and we can get some more B-roll shots with it. But guys, do be aware that if I do immediately go into a state of paralysis, just let the wasp fly away. Don't try to grab it, don't do anything. It'll be back in the wild and everything will be fine.
Right, Daddy.
This is in Arizona. This is like outside Tucson.
Million miles a second. This is the most nervous I've ever been to take a sting or bite from anything. My hand is shaking. Are you guys all ready?
Oh, yeah, I'm ready. You're ready.
I'm ready. Here we go. I'm Kylie Peterson, and I'm about to enter the sting zone with the tarantula hawk.
Go for it.
1, 2, here we go. 3.
You all right?
You all right?
Oh my gosh! Oh! Oh! Yeah. What are you doing?
All right, anyways. He goes on like that for a while. Anyways, watch the whole episode. It's hilarious. It starts off where he has to find the tarantula hawk. So he's like, him and his buddies are like going around like outside of Tucson with like a little bug catcher, and they finally found one. They catch it. Yeah, they're out here in Arizona. They're native. I've seen like maybe 4 in my entire life, and they're terrifying. Second most paid. Why'd that guy do that? Because he's an idiot. He's an absolute idiot. So anyways, second most painful sting. The first most painful, do you guys know? Bullet ant. The bullet ant. The bullet ant is the most painful insect sting out of all animals.
Wait, what is that?
It's an ant.
Is it a red ant?
I don't know what color it is. I think it's black. But bullet ant, it feels like literally, people say that it literally feels like getting shot by a gun. That's how painful it is. Yeah. So anyways, with pain and temperature, we're going to talk about different types of nociceptors. Remember, we talked about A beta. Now we're going to talk about A delta versus C unmyelinated fibers. So those are going to be your main nociceptors. We're going to talk about first and second pain. First pain is going to be the sharp pain that you feel right away. The second pain is what kind of lingers a little bit longer, and it's going to be more like a longer lasting dull pain. We're going to revisit some of these channels, the TRPV1. Remember, we discussed these when we talked about temperature, heat, capsaicin. Also, TRPV2 is going to be another one. There's going to be cold channel receptors. TRPA and TRPM are going to be your cold receptors. We're going to talk about the different pathways dorsolateral tract, anterolateral tract, dorsal column tract, part of the medial lemniscal system. We're going to talk about visceral and referred pain, which is especially important with organ systems, because if you have, like, for example, a heart attack, you're going to get referred pain. Right? Where do you get referred pain when you have a heart attack? Some of you guys might know. Down the left arm, shooting to the back, up the neck to the jaw. Right? That's the referred pain for the heart attack. You get referred pain from, like, liver conditions, from pancreas. You get gallbladder issues, bladder issues. You get referred pain that have distinctive patterns. So we'll talk about all of that. And then we're going to talk about the different structures that are all associated with pain sensation. So parts of the thalamus, the insular cortex, as well as the cingulate, gyrus, and cortex. And then also allodonia, which we talked about briefly before, but allodonia is where, like, even just the slightest thing can trigger severe pain. So it's part of, like, a neuropathy. Hyperalgesia, that's kind of like, hyperalgesia is like an umbrella term where you have extreme sensitivity to pain. And then we're going to talk about phantom limb pain as well as treatments for phantom limb pain. So good stuff. We're going to get into a lot of fun stuff with this section. So nociceptors, these are non-specific, right? This is not like your Merkel cells or Meissner cells or the Puccini cells, Puccinian or Ruffini cells, right? These are going to be just unspecific nerve endings. So these are going to be your free nerve endings. And there's going to be two types of fibers. One is going to be your A delta, and the second is going to be your C fiber. And your A delta are going to be myelinated, so they're going to be a little bit faster, whereas your C fibers are going to be unmyelinated, and they're going to be slower. They also have a smaller diameter. And then you get these nerves firing as soon as the noxious stimulus is enough to cause an action potential. Otherwise, you're not going to be able to perceive what's going on. And so here in this graph, they were subjecting an individual to different temperatures, and only at a certain point does the temperature get to where it actually begins to hurt. And so here is the temperature going up, and here is your nociceptors getting stimulated. And only around 43-ish degrees Celsius is when pain begins. Notice, 43 Celsius is 109.4 Fahrenheit. It's about to get to be that point here in Phoenix, right? So we're right around the corner to the upper hundreds. So June, July, when it's like really, really scorching hot outside, this is actually starting to stimulate your nociceptors. Right? Your body is actually perceiving that as painful stimulus. So hooray for Phoenix. Don't know why we live here. So here's the two major categories of pain. The first one that happens is going to be the first pain, and that's sharp pain, and it's transmitted by A delta fibers. And then the second pain is the one that lasts longer. It's going to be like a dull, delayed, achy type pain, and that's going to be your second pain, transmitted by your C unmyelinated fibers. There's also two subcategories of your A delta fibers. There's type 1. Those are going to be associated with potentially dangerous stimulus, like if you got stabbed by something, for example. Type 2 is going to be low heat threshold. So a little bit different. So two different subcategories. But broadly speaking, the important ones I want you guys to be familiar with: A delta, first pain, sharp pain, and C fibers, second pain, dull, prolonged. Yes?
So the type 2 is a low heat threshold because once you pick up that signal really fast, it's not going to be that same strength.
Low, okay, so high heat threshold, meaning that these get activated when you're maybe putting your hand on a hot skillet. It's like, oh, that's way too hot. So that means you have a higher heat threshold. Low heat threshold would be like, oh, it's 100 degrees. I'm starting to notice it. It's 105 degrees. I'm starting to notice it.
My brain thought they were connected for some reason.
What do you mean?
The faster one happens and then the delayed, like a process.
It kind of is a process. They're different nerve fibers, though. Yeah, they are connected, though. They're both going to get triggered by pain, but they can get selectively wiped out in experimental type settings. So if you did an experimental setting and you wiped out the A delta fibers, you're not going to experience that first sharp pain, right? You're only going to experience the prolonged, dull, achy pain that follows the noxious stimulus. Versus if you wipe out the C fibers, you're only going to experience that first sharp pain that's associated with your A delta fibers. Now, these are the different types of receptors associated with pain. You got your vanillinoid receptors. We talked about these before when we discussed capsaicin as well as temperature. These are your TRP receptors. So with like capsaicin, that was like TRPV1, right? Those are the ones associated with temperature, really hot stimulus, as well as capsaicin. Well, there's other TRPs as well. We'll go into those. You're going to find your A delta and C fibers associated with these. These are going to be ion channels. When they get stimulated, you get cations coming in. So sodium, calcium, for example. There's TRPM and there's TRPA. TRPM and A are going to be associated with cold. TRPM is specifically associated with a methanol type of feel. And this could be a painful stimulus, methanol. And then piezo. Remember your piezo family channels? Those are going to be those ones with the 3 subunits, right? They're like mechanoreceptors, right? So piezo 2s can also be associated with pain. And then there's an acid sensing channel. So ASIC is acid sensing, so you can detect changes in pH. When you get something like chest pain, for example, like if you have pectoral angina or if you're getting a heart attack, you know, these pain receptors are going to be responding to that, especially if you get changes in like pH, for example. One of the things that happens when you have chest pain and angina is you're not getting enough oxygen delivered to the heart, and so you're going to see like lactic acid buildup. So you're going to see lactic acidosis in those localized tissues. So revisiting the mechanism of capsaicin, remember that's going to be the active ingredient in your chili peppers. Those activate your TRPV1 channels. And so those are also going to respond to temperature changes. That's why you perceive pepper as being like heat, right? So capsaicin is basically affecting the same type of temperature channels. And the more you expose those receptors to this type of stimulus, the less sensitive they become. They become desensitized, right? That's why some people can't stand hot food, and then they never eat hot food. But like me, I was raised on really hot food, and I love it. So what's like not so hot to me might be really hot to, for example, you, right? Whereas some people can really experience super hot food, and it's like it doesn't faze them whatsoever. Like when I went to Mexico, what was it, Palenque and San Crispo de las Casas, like down in that region, like even the Pico de Gallo was really hot. And yeah, people there just ate it like no problem whatsoever. And I was like putting on my food and I was like, oh my God, this is insane. And I like hot food. I couldn't tolerate it. It was like really intense. But if you have more exposure to it, like you're less sensitive, which is another reason why you can use that to treat neuropathic pain, right? You can do capsaicin creams, or you could do a capsaicin patch, and that can help to reduce pain from neuropathy. So these are that's what the molecule looks like. Capsaicin, calcium, and sodium come into the TRBV channel. Right? Also heat would stimulate that as well. So we already talked about capsaicin before, so I'm not going to go into that in too much more detail. These are the major pathways. So you got the dorsal lateral tract. It's also referred to as the Lissauer tract. And then you have the anterolateral tract. The anterolateral tract is going to be associated especially with pain, temperature, and crude touch. So crude touch, rough touch, right? So that's anterolateral. And then you have the dorsal column medial lemniscus system, and that's going to be more like fine touch, pressure, vibration, proprioception. So dorsal column, more like the innocuous type of touch, whereas the anterolateral system, that's where you get actual noxious stimuli, pain, really hot or really cold. So let's watch a couple videos on these.
The pathway begins with nerve endings or receptors in the body, sending information down the axon of a neuron. These neurons are pseudo-unipolar neurons with cell bodies in the dorsal root ganglion, just like the ones we saw in the dorsal column medial lemniscus pathway. Now here's a slice of the spinal cord. And here is Lascelles fasciculus. The spinothalamic tract enters the spinal cord and ascends or descends 1 or 2 levels within Lascelles fasciculus. Then it synapses onto a cell body in the dorsal horn of the grey matter. The dorsal horns are the back parts of the butterfly wings of the grey matter. The axon from this neuron then decussates, which you'll remember means to cross to the other side, and then ascends via the spinothalamic fasciculus. This decussation is called the anterior white commissure. It is also important in the corticospinal tract, which we'll discuss in the next tutorial. These neurons here are located within the substantia gelatinosa, or the nucleus proprius, both of which are collections of cell bodies within the dorsal horn. The spinothalamic tract then ascends through the brainstem and synapses onto the thalamus. This is why the tract is called the spinothalamic tract, because it ascends from the spinal cord to the thalamus. The thalamus then sends projections to the post-central gyrus of the cortex. This is the same part of the cortex to which the dorsal column medial meniscus pathway projected.
All right, so you get the idea with that. So that's the major aspect of the spinothalamic tract. So that's dorsal lateral tract. Here's the anterolateral system. So this is pain.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss pain and the anterolateral system. Pain begins at nociceptors, cutaneous receptors that are specialized to detect noxious stimuli like extreme pressure, very hot or cold temperatures, or tissue damage. When activated, nociceptors send a signal to the spinal cord. The signal will be sent to the brain on pathways that make up what is known as the anterolateral system. The anterolateral system consists of 3 major pathways: the spinothalamic, spinal reticular, and spinal mesencephalic tracts. The spinothalamic tract can be considered the main pathway for transmitting pain information to the cerebral cortex. It is involved in the awareness and recognition of where in the body a painful stimulus is occurring. Neurons in the spinothalamic tract carry pain information from nociceptors to the spinal cord, then immediately synapse with a second neuron that quickly decussates or crosses over to the other side of the spinal cord before traveling up the spinal cord. These fibers proceed to the ventral posterior lateral nucleus of the thalamus, where they synapse with another neuron that carries the pain information to the somatosensory cortex. The function of the spinal reticular tract is not very clearly defined, but it may be involved in changes in the level of attention in response to pain. The fibers of the spinal reticular tract follow the same path as the spinothalamic tract, but they also project to areas in the reticular formation, which is a network of neurons found throughout the brainstem that are involved in a wide range of functions, including attention and consciousness. The spinal mesencephalic tract, also known as the spinotectal tract, plays an important role in the control and inhibition of pain. Fibers in the spinal mesencephalic tract follow the same path as the spinothalamic tract, but they synapse in an area of the midbrain called the periaqueductal gray. When neurons in the periaqueductal gray are stimulated, they can activate a natural pain suppression system that involves the release of endogenous opioids, which are substances that are produced by the body and have pain-killing effects. The release of endogenous opioids and other neurotransmitters leads to the inhibition of the pain signal in the spinal cord and naturally produced analgesia.
All right, so anterolateral system. Now let's see the medial meniscal system.
Welcome to 2-Minute Neuroscience.
Oh, wait, we already watched that from the last one, so never mind. We already covered the medial meniscal system from the last section on pressure, vibration, proprioception. Yeah.
Sorry, I have a question. But I remember you said that the anterolateral system has to do with the actual pain stimulus that you said.
Uh-huh.
But what was the thing you said about the dorsal column medial meniscal system?
Oh, so anterolateral, think about that as being all of your noxious stimulus. Pain, temperature, crude touch, right? Things that are unpleasant. Versus dorsal column, medial meniscus, that's going to be just regular touch, right? You give somebody a high five, unless it's a really intense high five. If it's a really intense high five, that'll be carried by the anterolateral system.
So no pain, basically?
Yeah, exactly. So dorsal column is going to be just proprioception, vibration, just standard sensation. So here's you're welcome. So anterolateral system. So again, these come in from the dorsal horn. You got your pseudo polarized neuron, right? And then you're going to see they're entering in through Lissauer's tract until they enter in and then decussate across the other side of the spinal cord until they go all the way up to the thalamus. C fibers, these are going to be associated with Rex lamina. You guys remember Rex lamina from, I think, week 1? Right? You got all these different Rex lamina within the spinal cord. We only talked about the nucleus gelatinosa, which is Rex lamina 2, right? Huh? It was like, we talked about 2 and 9. Yeah, those are the major ones. But now we're going to talk about some other ones as well. So C fibers, that's going to be your like slow pain, second pain. Right? That's going to be associated with Rex lamina 1 and 2. Some are going to be associated with 5. But then your A delta, which is your fast first pain, that's going to be also associated with Rex lamina 1, but also 5 as well. So this is going back to week 1 when we talked about Rex lamina. So last time I only wanted you guys to know your substantia gelatinosa, which was associated with pain transmission. But here are the other regions now too. So you got your anterolateral system associated with Rex lamina 1 as well. Anterolateral is also associated with Rex lamina 5. You also have, what else here? Interneurons associated with the intermediate gray. That's not super high yield for this. Then you have your motor columns, which is associated with 9. So that was like a review from week 1 of our Rex lamina. So now the major ones that we included here now are going to be Rex lamina 1 and Rex lamina 5, basically. 1 is associated with anterolateral as well as Rex lamina 5, anterolateral system. So here's your anterolateral system and your dorsal column medial lemniscus system. Dorsal column medial lemniscus system gets all the way to the medulla until it decussates, right? So it's going to go up on one side ipsilaterally until it decussates at the medulla. So in other words, if you have a lesion in your spinal cord, you're losing sensation of fine touch and proprioception, you're going to lose fine touch and proprioception on the ipsilateral side of the lesion. The anterolateral system is going to decussate at the level of the spinal cord. And so if you had a lesion to the spinal cord, you're going to lose pain and temperature to the side, the other opposite end of where it decussates. So here's your differences in pain and temperature versus vibration and proprioception. So here's a lesion at T10. So if you had a lesion, this is at the lumbar spine, and you have loss of sensation at that region, you're also going to see the lower appendages having some interesting changes as well. So if you see anterolateral system, which carries pain and temperature, that's going to be on the contralateral side. You're going to lose that sensation on the opposite side of the lesion, versus dorsal column is going to be on this ipsilateral side of the lesion. I don't know if I explained that too well. Regardless, it's the end of class. So this is where we're going to continue next time. I'll go into more detail about this because this is something that will probably wind up on your exam. So what kind of sensations you lose depending on the side of the lesion. Cool. Any questions? Awesome. Sounds good. So next time we meet, we'll continue there, and then we'll get into vision as well. So right on, guys. Have a good one.